Today, I will talk about oncolytic viruses in general, and I will spend the last 10 minutes of my presentation talking about my work. The title that I gave is really a take-home message. And the take-home message is that viruses are not only the devastating agents of pandemics, but we can actually manipulate viruses and make them our friends, and we can turn them into anti-cancer weapons. So let's start with a definition. What are oncolytic viruses? Oncolytic viruses exploit the intrinsic ability of almost any virus to kill the infected cell, apart from transforming viruses. The lytic viruses bring the infected cell to death. And they, of course, they bring to death also cancer cells. And the unique property of oncolytic viruses as anti-cancer agents is that the viruses multiply and spread to adjacent cells. So essentially, the quantity of the drug that we administer increases with time. And the oncolytic viruses have been derived from different virus families. So this is something I will cover. And they were derived both from human viruses or from animal viruses. Initially, they were derived from human viruses. Human viruses need to be modified because you don't want them to be pathogenic. But there is also one problem, that humans have antibodies to these viruses. And so there is a prior immunity when we use these oncolytic agents based on human virus. And the problem, prior immunity will neutralize somehow the drug that we administer. The main human viruses that have been converted to oncolytic viruses are adenoviruses, herpes simplex viruses, measles virus, and reoviruses. And then in order to overcome the problem of the prior immunity, animal viruses capable to infect human cancer cells have been introduced as oncolytic viruses. And this list includes pox viruses derived from cowpox, pox virus, vesicular stomatitis virus, and other animal virus of human origin. So what is the historical background of using viruses as oncolytic agents? It was reported in the scientific literature that occasionally cancer patients who contracted an infectious disease underwent brief periods of clinical remission. And this occurred mainly with leukemia and lymphoma. And this is a paper published in Lancet in the 50s, where you can see that a boy affected by a lymphoma, he acquired measles, and for a short period, the lymphoma regressed. So the notion that viruses may be turned into anti-cancer agents was pioneered in the early 90s by Roseman and by Bertuzza. Bertuzza is actually a neurosurgeon in Harvard, and they both derived the idea separately, actually, that viruses can be turned into oncolytic viruses. And this idea, cumulative idea, came from three, actually, pillars. First, it is well known that viruses grow better in cancer cells, the famous, for example, HeLa cells, than in non-cancer cells. And the reason is that cancer cells carry variable defects in the innate response to the virus. So they pose less obstacles to virus growth. And in those years, the genetic engineering technologies to modify viruses had been just established. And there was obviously a clinical need to be met. Cancer, as of today, is the second most frequent cause of death, severely affect the patient's life. Chemo and radiotherapy are toxic, greatly impact on the quality of life of the patients. And there are some cancers we still have a very high mortality rate. They are almost death sentences. And these are the metastatic cancers, glioblastoma, cancer of pancreas. There are cancers which have not improved in all these years, in recent years. So the rule number one, if you want to design an oncolytic virus, is that you need to kill the tumor and spare the patient. And it cannot be vice versa. You don't want to kill the patient and have the tumor surviving. And in order to do this, you have to modify the viruses in order for them to be cancer selective, or, as we try to do nowadays, to be cancer specific. So selectivity, the majority of the oncolytic viruses which are in clinical trials are cancer selective. Selectivity rests either on the property that I just mentioned, that cancer cells pose less obstacles, less innate immunity to viral growth than non-cancer cells, or may result from genetic modification, selection of mutants, selection of viral viruses. And all this we call attenuation of the virus ability to replicate. The attenuated oncolytic viruses are attenuated in the way they are weaker viruses, but they can still replicate in the cancer cells. And they are more or less severely hampered in their ability to replicate in the non-cancer cells. The extent of infection in non-cancer cells causes toxicity, and this is clear. So there is actually an equilibrium between safety, which results from attenuation, or efficacy. Efficacy results from having a virus which keeps its virulence. Historically, if you look back to what happened, safety was the dominant concern. So the viruses which are actually now in clinical trials are attenuated and sometimes even over-attenuated. My interpretation is that actually the oncolytic virus research started at the same time as gene therapy employment of viruses. And for the gene therapy, there were accidents. So safety was really a concern. And I have to say that the oncolytic viruses are really very well tolerated. There has never been an accident. Of course, the patient is different. And so this has to do. But so anyway, there is an equilibrium. If you want to stay on the safe side, you go for attenuation or over attenuation. But now we recognize that these viruses as monotherapy are not so effective. And so we want to actually increase efficacy. To put the oncolytic virus into frame, there is so far only one oncolytic virus which has been approved. And this is a partially attenuated herpes simplex virus named Defect or Oncovec GM-CSF. And it has been approved only against one indication, melanoma. The next in line for approval will come, it is expected soon, will be another herpes virus against glioblastoma. So let's look now what viral species have been converted into oncolytics. This is a rather busy slide, but I will go through it. So oncolytic viruses were derived both from DNA viruses. DNA viruses are the viruses whose genome is a molecule of DNA, or RNA viruses. RNA viruses are the viruses whose mini chromosome is an RNA molecule. And here I have listed some of them, the most important ones. And any virus is different, so any virus has different advantages and disadvantages. And I have listed here for you the major pros and cons. Let me start from herpes simplex virus, which is my pet virus. So herpes virus is a rather big virus. The genome is 150 kilobase pair. So this is an advantage because there is a space for insertion in the virus when we use it as an oncolytic virus for extra transgenes, for cellular genes, which can modulate the activity of the oncolytic virus. I will come to this. So the genetic engineering is feasible. You just have to learn how to do it. And tropism retargeting, which is what I personally do, is actually feasible. And the reason is that herpes virus is not depicted, but as an envelope, and the glycoproteins, which make the envelope, are prone to modifications, much more than viruses which have a capsid. Herpes have another advantage, and there is a specific drug which can be employed on worst case scenario. This is a cyclovir or ganciclovir. And you can see that no other virus has actually this important property. And all the viruses populate the tumor, so it will populate them. The con is that there is prior immunity in the human population. Now, if we look, for example, to adenoviruses, which are the most frequent oncolytic viruses now in clinical trials, they still have a genome reasonably big enough that it can host some transgenes. Genetic modification can be achieved. Tropism retargeting is very difficult because the capsid is very much constrained. There is no antiviral drug, and there are antibodies in the human population. And this is why also, for example, in the COVID vaccine, they are using adenoviruses derived from animals, monkeys, for example. Vaccinia virus is a much bigger virus, so the genetic modifications are not so easy to perform. The tropism retargeting has not even been tried. And the population, people old as I am, they were vaccinated until the 80s. The children were vaccinated again the vaccinia virus, yes. But the young population, there are no antibodies. Concerning the RNA viruses, an interesting oncolytic virus is the measles virus derived OV. So, but you can see here, it's immediate, it comes to your eyes that the genome of these viruses is rather small. So if you want to introduce an extra gene, you have to delete one of the viral genes. So, and at best, you can put one transgene. For measles virus, the genetic modification are readily done. The tropism retargeting can be done. There is no drug. And the virus, which is used as oncolytic for measles virus, is actually the virus that is used to vaccinate people. So essentially, most of the population has antibodies to measles virus, and this is a problem. And for example, NDV is an animal virus. The animal viruses were introduced in order to avoid the problem of the prior immunity. So let's see how these viruses perform in clinical trials. Herpes virus is the only one the only family for which an oncolytic virus has been approved. The virus which is approved carry a transgene, which is GM-CSF, so granulocyte monocyte colony stimulating factor. It is deleted in a gene. I will explain why it is deleted. The next most important are adenoviruses. They are in phase 2, in phase 3. They behave well, and that's it. They also host heterologous genes. The pox virus looked very promising because it can also be administered systemically, which is not listed here. However, in 2020, this virus failed twice in phase 3, so now it's really a problem. It failed in Europe and it failed in South Korea. Now, for example, the RNA viruses, I mentioned the measles viruses, the virus which is used is the vaccine strain, so it is highly safe because millions of individuals have been vaccinated. Of course, there is a problem of prior immunity. And then there is the bunch of animal viruses, which replicate in human cells and replicate better in tumor cells than in non-tumor cells. And a reovirus, which is called the rheolizine, has actually the advantage that it can be administered systemically. So I want to give you one example of how we attenuate virus. I cannot cover attenuation of all the different viruses. So I will give the example of herpes simplex virus. Most of the oncolytic viruses based on herpes simplex, including the one which is approved, carry the deletion of a gene which is called the 34.5 gene. Let's see how it works. So I have to give for the young people, if they are listening, a small background, really small, on innate immunity. The cell is equipped to contrast the virus. When a cell is infected by the virus, the cell reacts, counteracts the infection. And this is done through a response, which is usually called the innate response. And there are several sensors in the cell which detect the presence of a virus. And the sensors, in case of HSV, are the toll-like receptors. We worked at this, actually, we discovered this. And there are other cytoplasmic sensors, gas, sting, IFI-16, and so on. And FKB becomes activated. All this goes to activate some cellular genes, among which is interferon. Interferon is the most active response that the cell put in place against the virus. It's an arm against the virus, but it's not only interferon. Interferon is here for simplicity, but as I will show to you, there are a number of cytokines, chemokines, other factors. So, interferon will, and double-stranded RNA, will activate a protein in the cell, which is called protein kinase R. And what protein PKR does is to phosphorylate a translation factor, EIF2 alpha. And when EIF2 alpha is phosphorylated, the protein synthesis is blocked. In this case, the protein synthesis, most of the cell and of the virus are blocked. So the cell senses herpes, activates PKR, and blocks protein synthesis, actually destines itself to death. Of course, viruses, as we know, are very smart. So the virus has a strategy to counteract, to evade this cellular activity. How does it work? So PKR phosphorylates EIF2 alpha. There is a block in protein synthesis. But the virus has a gene, which is the 34.5 gene. And this gene product brings a phosphatase to EIF2 alpha, which becomes dephosphorylated, and the protein synthesis is restored. So this is the way in which the virus evades the cell defense. So it was discovered in the 90s. Cancer cells have a defect in PKR activation, so cancer cells do not activate this pathway. And the virus can replicate a virus deleted in 34.5, can replicate well because the virus doesn't, the cancer cells do not have the PKR. So discoveries were that HSVs deleted in the 34.5 gene grow fine in cancer cells. They are impaired, of course, to some extent in non-cancer cells, which are PKR positive. And this is actually the base of selectivity for the OVs that are cancer selective, that are deleted in the 34.5 gene. So, how do oncolytic viruses exert their anti-cancer activity? This is something which is, has come out in recent years. The oncolytic virus causes the cell to die. But actually the death is very interesting because the viruses cause what is called immunogenic cell death. So the viruses which have been chosen to become oncolytic viruses, they cause a type of cell death which will stimulate the immune response. And this is done through modification to the tumor microenvironment. Essentially, there are cell debris which are released. There is a whole bunch of modification to the tumor microenvironment, which I will show to you. And we can actually augment this through transgenic expression of proteins. And so this will bring a defense at the level of the tumor that we treat with the oncolytic virus. But all these changes to the tumor microenvironment and this activation of the immune response actually vaccinate the individual against the tumor. And so there is a defense also against distant tumors. And this is better seen here. When the cancers, and this is why the oncolytic viruses now are better termed as oncoimmunotherapeutic viruses. The tumor cell, when infected by oncolytic viruses, the cancer cell die of immunogenic cell death. There are a bunch of modifications to the tumor microenvironment, which result in activation of CD4, CD8 cell, and K cells. And these actually are recruited to the tumor microenvironment, to the tumor bed. And now there is an enhanced local and systemic anti-tumor activity. So please memorize this. Intratumorally, the virus causes immunogenic cell death. The virus causes what is called an immune heating of the tumor microenvironment. The tumor microenvironment is highly immunosuppressive. And now the tumor microenvironment, once it is infected by the OV, is not any longer so immunosuppressive. And this will bring to activation of an anti-cancer T-cell response, both local and systemic. This is done actually through secretion of a number of cytokines and chemokines. I don't want to go into it. These are the infected tumor cells. In the tumor bed, the monocyte, macrophages, dendritic cell also in turn secrete all these factors. This will activate, as I mentioned, the T cells, memory and naive, and this cell will populate the tumor. So, secretion of IFN, activation of NK cells, of dendritic cells, recruitment and activation of lymphocytes. I want to open a parenthesis here. This is actually the same thing as happens in COVID, because the cell has a response to the virus, a defensive response. I think we all know that the first days of coronavirus 2 infection, there is viral replication, and actually the individual can be asymptomatic or paucisymptomatic. But the real problem of COVID comes 5 to 7 days after the infection, when actually in the lung tissue, there is ongoing this secretion of all these cytokine and chemokines. For example, you remember that there has been also in Italy a clinical trial finalized to block IL-6. So essentially what I want to emphasize is this is a common response of the infected cell to the virus. When this occurs in a tumor cell, then it will defend us from the tumor. But during an infectious disease, it may actually be part of the pathogenesis of the disease. This is just a small parenthesis. So we can actually potentiate the immunotherapeutic activity of the oncolytic viruses, and we can do it in two ways. Either we can arm the oncolytic viruses by genetically engineering in the genome of the oncolytic virus immunostimulating molecules. In this case, the bigger the viral genome, the better. HSV are ideal for this aim. Or we can combine with the checkpoint inhibitors. So arming is genetically engineering in the viral genome a bunch of immunostimulatory genes. Some are reported here, GM-CSF, IL-2, IL-12, IL-15, and so on. Or for those of you who don't have a background on checkpoint inhibitors, I have this slide. Checkpoint inhibitors were discovered mainly by Ellison and by Kono. They received the Nobel Prize for this discovery in 2018. And the very basic story is the following. When a T lymphocyte interacts with the antigen-presenting cell or also with the tumor cell, of course, there is T cell receptor, MHC, but there are also inhibitory pathways. So the immune response is inhibited. For example, is inhibited by the axis PD-1 ligand, or from the CTL4, or from other blockers. And the important finding from Ellison and Kono was that if you actually put antibodies to CTL4, to PD-1 ligand, you release this inhibition and you restore the immune response. So very schematically, if a T cell reacts with the tumor cell in the presence of the PD-1 ligand, there is no way that the T cell can inhibit this response. But if you administer the antibody to PD-1 or PD-1 ligand, the T cell will exert the inhibitory response. And so will inhibit tumor growth. Unfortunately, there are limitations to these checkpoint inhibitors. And the major limitations are that checkpoint inhibitors are active only against a limited number of tumors and against a limited number of patients. So there is a need worldwide to increase the spectrum of activity of checkpoint inhibitors. And all these are really the ideal partners of checkpoint inhibitors, because we modify the tumor microenvironment, as I just showed to you, and they release part of the immunosuppression of the tumor and greatly increase the efficacy of checkpoint inhibitors. And now I would like to switch to a few slides of my work. So our approach actually started from my background on how HSV entered the cells. And our approach was, we didn't want to make an attenuated virus. We wanted to leave the virus unattenuated. I'm talking about herpes simplex virus. And in order to have an oncolytic virus, which is unattenuated, the approach that we took was to engineer an oncolytic virus that infects tumor cells only, or possibly only. So we wanted to go for a virus which is cancer specific and not the cancer selective. And the line of research that we followed was to modify the tropism of the virus and retarget the tropism of the virus to the cancer cells, which is depicted here. This would be the wild-type virus, which is its glycoprotein, here the blue. The wild-type virus glycoprotein interact with the natural receptor of the virus, which is also blue, and this leads to HSV entry into the cell. Now we modified the glycoproteins. The major glycoprotein that we modified was GD, but not only GD. So we modified the glycoprotein. Let's talk about glycoprotein D. So as the glycoprotein does not any longer interact with the wild receptor of the present in the wild-type cell, in the normal cell, but it will interact with the molecule present on the cancer cell. And we want to transform this molecule present on the surface of the cancer cell as the portal of entry of the virus. And actually this works. So we modify the glycoprotein so that it interacts with a selected protein present on the cancer cell plasma membrane. And now this protein will become the portal of entry of the virus. The virus is rather complicated in terms of entry. It has 4 essential glycoproteins, which act in a cascade manner. There is a cascade of activation. The glycoprotein D interacts with the receptors, the GD receptors. The receptor activated D will activate GH-GL, remove GL, interacts with the integrin. All this leads to activation of glycoprotein B, which is the glycoprotein which actually mediates the fusion of the virion envelope with the plasma or endocytic membrane. So when we retarget the virus, glycoprotein D is not any longer able to react to the natural receptors. It will react to the receptor that we have selected, which in our case most frequently is HER2, and then there is the cascade of activation, which culminates with the fusion of the virion envelope with the cell membrane. So this is our first fully retargeted virus. This is the genome of HSV, 150 kilobase pair. This is the GD gene, which is here. Initially, we made a deletion. This deletion removes the ability to interact with the natural receptors. And in place of this deleted region, we inserted a ligand to HER2, which was a single chain antibody to HER2, that you can see here on the left side. We deleted the red portion, which is the amino terminal portion of GD. And now in this deleted GD, we inserted a single chain antibody, which is actually almost as big as GD. And this chimeric glycoprotein now actually really retarget the virus tropism. The virus is not any carrying this chimeric GD, is not any longer able to use the 2 GD receptors, Nectin-1 and HVEM, as a portal of entry, but it will use HER2 as the portal of entry. And with this virus, we can actually cell culture show that it only infects cells positive for HER2, tumor cells HER2 positive. It does not infect the tumor cells HER2 negative. It happens that it also infects 293 cells and doesn't infect a bunch of other cells. So this tropism retargeted oncolytic herpes are tumor specific. Of course, they are tumor-specific to the extent that the molecule that we select is tumor-specific. But they do not carry any genetic modification in virulence or any other gene except the glycoprotein. The safety of these viruses rests on the specificity, not on the attenuation. And once these viruses infect the cancer cell, they are actually fully virulent virus. So our design was to have a virus which in the cancer cell is really strong, robust, and whose safety relies on the specificity. The target that we have studied more is the human HER2, which is present in a number of tumors, breast, ovary, lung, stomach, pancreas. But we can actually, with the same technology, retarget to other receptors like prostate, PSMA, the EGFR present in glioblastoma, and so on. So we think that this actually is a platform. So the first experiments were done, of course, as it happens, in nude mice implanted with the human cancer cells positive for HER2. And here in this model, we provided the first proof of principle of anti-tumor efficacy. Here you see the growth, the kinetics of tumor growth. If the mice receive one single injection of the virus that I showed to you, the tumor growth is very much inhibited. Actually, it works very fine in the nude mice. And this is the peritoneum of a mouse in which we administer intraperitoneally the tumor, and then we administer the virus. The virus carries a GFP. So all the tumor masses are green, but there is no green in the organs of the peritoneum other than in the tumors. And here you can see the peritoneum of untreated mouse. These are the tumor masses. And this is the peritoneum of a mouse which was treated with the oncolytic, first generation oncolytic virus. And we proved efficacy also in another model, in the glioblastoma model. Here the tumor is implanted in the brain, the tumor carries a red GFP. When it is treated, the tumor does not appear. So I want to go to my last 3 slides. And I want to show you the efficacy of our first viruses that we call the first generation, and the last viruses that we generate, which are the 3rd generation. First generation is 133. The 115 virus carries IL-12. And the 3rd generation virus also carries an IL-12. And this is the experimental model. We administer the tumor subcutaneously, in this case. When the tumor is established, we administer the virus. If the virus carries IL-12 in the tumor bed, we will have a secretion of the factors, IL-12, interferon, GM-CSF, TNF, activation of the anti-tumor immune cells, CD8 positive, CD4 positive, NK cells, and all this will cause a reduction in the primary tumor. But it also induces a vaccination against tumor. So now, when we administer a second tumor distally, and we don't treat the challenged tumor, the challenged tumor does not grow because the virus carries a T-cell response against the tumor. And I want to show you the evidence for this. These are the first generation virus. The virus which does not have IL-12 actually behaves, is very modest. When IL-12 is inserted, there is a delay in tumor growth. And when we administer the third generation virus, you can see that the tumor grows and then the tumor regresses, and we have a complete response in 8 out of 10 mice. Now we take the mice which survived the primary tumor. These mice here, which did not die, and these mice here. And we implant later a challenge tumor. And we can see that the challenge tumor does not grow anyway. So the mice are fully protected. The mice which survived the primary tumor are fully protected from a challenge tumor. So we really have an immunotherapeutic agent, not only an oncolytic virus, but also an immunotherapeutic agent. These are all the changes that we have, let's say, measured in the tumor microenvironment, increase in CD8 cell, in NK cells, in CD4 cells, in the dendritic cells, and in the number of factors. Now, do our viruses synergize with the checkpoint inhibitors? Yes. You can see that this is a tumor treated with the anti-PD-1, with the checkpoint inhibitor. You can see that the checkpoint inhibitor as a monotherapy does nothing. So this is the example of a tumor which really is resistant to checkpoint inhibitors. Now we give our virus a smaller amount than I showed before. So we protect only some mice, and then, but in the combination treatment, we have a much higher effect. So with the monotherapy of 337 at this dose, these are the mice, 40% of the mice are protected. If we combine the two treatments, we have protection in 80% of the mice. And I go to my last slide, which is the following. Can this virus, which is so robust, actually be administered systemically? The answer is yes. In this case, the tumor cells are implanted via the tail vein, and they cause tumors in the lungs. Each one of these nodules is a tumor. It's a model, it's considered to be a model of metastatic tumors. If we treat with our virus, these are the lungs. So they are spared or there is a decrease, and this is quantification of the decrease. So the efficacy upon systemic administration of an oncolytic virus has been documented rarely, and I think it can be considered a very good result. So the conclusion for my part of the work on oncolytic virus is that when the oncolytic virus is armed with IL-12, we see good inhibition of the growth of primary tumors, an immunotherapeutic vaccine effect, combination with checkpoint inhibitors, efficacy upon systemic administration. I did not touch the problem of manufacture, which we have addressed. So these were the people with whom I have been collaborating here in the University of Bologna, the Beta group again in the University of Bologna, Malatesta for the University of Genoa for the glioblastoma. I also initiated the collaboration with the company. Now the collaboration is terminated, and I thank you for listening.